Maximizing the lifespan. Chapter 1. Live a longer life. The maximum lifespan at en.wikipedia.org/wiki/maximum_life_span. Maximum lifespan, or, for humans, maximum reported age at death, is a measure of the maximum amount of time one or more members of a population have been observed to survive between birth and death. The term can also denote an estimate of the maximum amount of time that a member of a given species could survive between birth and death, provided circumstances that are optimal to that member's longevity. Most living species have an upper limit on the number of times somatic cells not expressing telomerase can divide. This is called the Hayflick limit, although this number of cell divisions does not strictly control lifespan. In animal studies, maximum span is often taken to be the mean lifespan of the most long-lived 10% of a given cohort. By another definition, however, maximum lifespan corresponds to the age at which the oldest known member of a species or experimental group has died. Calculation of the maximum lifespan in the latter sense depends upon the initial sample size. Maximum lifespan contrasts with mean lifespan, average lifespan, life expectancy, and longevity. Mean lifespan varies with susceptibility to disease, accident, suicide, and homicide, whereas maximum lifespan is determined by rate of aging. Longevity refers only to the characteristics of the especially long-lived members of a population, such as infirmities as they age or compression of morbidity, and not the specific lifespan of an individual. The longest living person whose dates of birth and death were verified according to the modern norms of Guinness World Records and the Gerontology Research Group was Jean Calment, 1875–1997, a French woman who is verified to have lived to 122. The oldest male lifespan has only been verified as 116, by Japanese man Jiroemon Kimura. Reduction of infant mortality has accounted for most of the increased average lifespan longevity, but since the 1960s mortality rates among those over 80 years have decreased by about 1.5% per year. The progress being made in lengthening lifespans and postponing senescence is entirely due to medical and public health efforts, rising standards of living, better education, healthier nutrition, and more salubrious lifestyles. Animal studies suggest that further lengthening of median human lifespan as well as maximum lifespan could be achieved through calorie restriction mimetic drugs or by directly reducing food consumption. Although calorie restriction has not been proven to extend the maximum human lifespan as of 2014, results in ongoing primate studies have demonstrated that the assumptions derived from rodents are valid in primates. Studies in the biodemography of human longevity indicate a late life mortality deceleration law, that death rates level off at advanced ages to a late life mortality plateau. That is, there is no fixed upper limit to human longevity, or fixed maximal human lifespan. This law was first quantified in 1939, when researchers found that the one-year probability of death at advanced age asymptotically approaches a limit of 44% for women and 54% for men. However, this evidence depends on the existence of a late life plateaus and deceleration that can be explained, in humans and other species, by the existence of very rare errors. A theoretical study suggested the maximum human lifespan to be around 125 years using a modified stretched exponential function for human survival curves. In another study, researchers claimed that there exists a maximum lifespan for humans, and that the human maximal lifespan has been declining since the 1990s. Lifespan in animals. Small animals such as birds and squirrels rarely live to their maximum lifespan, usually dying of accidents, disease, or predation. The maximum lifespan of most species is documented in the Age repository, the Animal Aging and Longevity Database. Maximum lifespan is usually longer for species that are larger or have effective defenses against predation. The records, in years, are these: common house mouse, 4. Brown rat, 3.8. Dogs, 31. Cats, 38. Common cranes, 43. Polar bears, 42. Horses, 62. Asian elephants, 86. Large parrots, macaws and cockatoos can live up to 80–100 years in captivity. Koi, a Japanese species of fish, allegedly living up to 200 years, though generally not exceeding 50. A specimen named Hanako was reportedly 226 years old upon her death. Tortoises, Galapagos tortoise, 190 years. Tuatara, a New Zealand reptile species, 100–200 plus years. Eels, the so-called Brantivik eel, Swedish, Brantiviksälen, is thought to have lived in a water well in southern Sweden since 1859, which makes it over 150 years old. Whales, bowhead whale, Balana mysticetus, about 200 years. Lobsters are sometimes said to be biologically immortal because they do not seem to slow down, weaken, or lose fertility with age. However, due to the energy needed for molting, they cannot live indefinitely. Caloric restriction in life extension. Rats, mice, and hamsters experience maximum lifespan extension from a diet that contains all of the nutrients but only 40–60% of the calories that the animals consume when they can eat as much as they want. Mean lifespan is increased 65% and maximum lifespan is increased 50%, when caloric restriction is begun just before puberty. For fruit flies, the life-extending benefits of calorie restriction are gained immediately at any age upon beginning calorie restriction and ended immediately at any age upon resuming full feeding. Ideas to extend life. Most biomedical gerontologists believe that biomedical molecular engineering will eventually extend maximum lifespan and even bring about rejuvenation. Anti-aging drugs are a potential tool for extending life. Aubrey de Grey, a theoretical gerontologist, has proposed that aging can be reversed by strategies for engineered negligible senescence. De Grey has established the Methuselah Mouse Prize to award money to researchers who can extend the maximum lifespan of mice. So far, 3 mouse prizes have been awarded, one for breaking longevity records to Dr. Andrei Bartka of Southern Illinois University, using GHR knockout mice, one for late-onset rejuvenation strategies to Dr. Steven Spindler of the University of California, using caloric restriction initiated late in life, and one to Dr. Z. Dave Sharp for his work with the pharmaceutical rapamycin. DNA damage theory of aging. Accumulated DNA damage appears to be a limiting factor in the determination of maximum lifespan. The theory that DNA damage is the primary cause of aging, and thus a principal determinant of maximum lifespan, has attracted interest in recent years. This is based, in part, on evidence in humans and mice that inherited deficiencies in DNA repair genes often cause accelerated aging. There is substantial evidence that DNA damage accumulates with age in mammalian tissues, such as those of the brain, muscle, liver, and kidney. One expectation of the theory that DNA damage is the primary cause of aging is that among species with differing maximum lifespans, the capacity to repair DNA damage should correlate with lifespan. The first experimental test of this idea was by Hart and Setlow, who measured the capacity of cells from 7 different mammalian species to carry out DNA repair. They found that nucleotide excision repair capability increased systematically with species longevity. This correlation was striking and stimulated a series of 11 additional experiments in different laboratories over succeeding years on the relationship of nucleotide excision repair and lifespan in mammalian species. In general, the findings of these studies indicated a good correlation between nucleotide excision repair capacity and lifespan. The association between nucleotide excision repair capability and longevity is strengthened by the evidence that defects in nucleotide excision repair proteins in humans and rodents cause features of premature aging. Further support for the theory that DNA damage is the primary cause of aging comes from study of poly-ADP ribose polymerases, PARPs. PARPs are enzymes that are activated by DNA strand breaks and play a role in DNA base excision repair. For mammals, there is a direct relationship between mitochondrial membrane fatty acid saturation and maximum lifespan. Studies of the liver lipids of mammals in a bird, pigeon, show an inverse relationship between maximum lifespan and number of double bonds. Selected species of birds and mammals show an inverse relationship between telomere rate of change, shortening, and maximum lifespan. Maximum lifespan correlates negatively with antioxidant enzyme levels and free radicals production and positively with rate of DNA repair. Female mammals express more MN-SOD and glutathione peroxidase antioxidant enzymes than males. This has been hypothesized as the reason they live longer. The maximum lifespan of transgenic mice has been extended about 20% by overexpression of human catalase targeted to mitochondria. A comparison of 7 non-primate mammals, mouse, hamster, rat, guinea pig, rabbit, pig and cow, showed that the rate of mitochondrial superoxide and hydrogen peroxide production in heart and kidney were inversely correlated with maximum lifespan. A study of 8 non-primate mammals showed an inverse correlation between maximum lifespan and oxidative damage to mtDNA, mitochondrial DNA, in heart and brain. A study of several species of mammals and a bird, pigeon, indicated a linear relationship between oxidative damage to protein and maximum lifespan. There is a direct correlation between DNA repair and maximum lifespan for mammalian species. Fat-specific insulin receptor knockout, FRCO, mice have reduced fat mass, normal calorie intake and an increased maximum lifespan of 18%. The capacity of mammalian species to detoxify the carcinogenic chemical benzo(a)-pyrene to a water-soluble form also correlates well with maximum lifespan. Short-term induction of oxidative stress due to calorie restriction increases lifespan in Caenorhabditis elegans by promoting stress defense, specifically by inducing an enzyme called catalase. As shown by Michael Risto and co-workers, nutritive antioxidants completely abolish this extension of lifespan by inhibiting a process called mitohormesis. Telomeres shorten more slowly in long-lived birds and mammals than in short-lived ones.